Good afternoon, everyone. Welcome back to Virology. So for the next two lectures, we're going to talk about how host organisms respond immunologically to a virus infection. Obviously, two lectures is not enough to cover immunology, but it's enough so that you understand how viruses are dealt with. So today, and there are two sections. The first is innate and intrinsic and innate defenses. We're going to talk about that today. Next time, adaptive defenses. So this is my view of the whole immune system. We have four sets of barriers. The first are anatomical and chemical barriers, like the outer layer of your skin, right? The mucus layers on your respiratory and gastrointestinal tract, et cetera, tears, defenses on your skin, et cetera, even saliva. And that's always around, right? So it's always there to protect you. Many viruses get trapped in mucus and never get anywhere. Then we have intrinsic defenses, which are also around all the time. They're available immediately, and we'll talk about those. They include interferons, autophagy, apoptosis, microRNAs, and CRISPRs. Then we have the innate immune system, which needs from minutes to hours to get activated once a pathogen is in. And this includes natural killer cells, complement, antigen-presenting cells, neutrophils, and cytokines. And then adaptive immunity, which we'll talk about next time, comprising B and T cells, and memory. So viruses encounter each of these barriers. And if they get over one, they encounter the next and the next and the next and so forth. And if they get over the final barrier, acquired immunity, then you have some kind of disease. So here are the host defenses in another way. Intrinsic are always present in the uninfected cell, and I've given you some examples already, right? Intrinsic is ready to go, and the innate immune system is induced by infection. It needs some warming up, and the adaptive is tailored to the pathogen. That's probably the best way to distinguish the two, is that it's tailored to the pathogen, so it takes longer to develop. So some examples of intrinsic defenses: RNA interference. This is largely important in plant and invertebrate cells. In mammals, there's a controversy over whether they're present or not, and if they're needed, because we have a protein-based defense system, and many other organisms do not. So in a RNA interference, a virus comes in to the cell, and viral double-stranded RNA is part of its reproduction cycle. An enzyme called dicer from the cell chops it up into small pieces, double-stranded pieces. One of the strands is removed, and that's called the siRNA, small interfering RNA, and that is linked up with a RNA-induced silencing complex, which includes AGO, Argonaut, and then this RNA will be complementary to other viral RNAs in the same cell, so it will bind to it. So that RISC complex will bind to the viral RNA and chop it up. So that's an intrinsic defense that's always present. We have siRNA, we have microRNAs. We produce them in germ cells in particular, but it's just not clear if mammals need it, because we have so much else. We certainly use it to regulate gene expression, but for virus infections, it's not clear. It's not been clear for a number of years now, so I'm waiting for it. I'm waiting for clarity, but I don't see it. So I would say right now, plants and invertebrates are the main beneficiaries. Now here is another intrinsic defense. This is a protein called ApoBec3. And this stands for apolipoprotein B, mRNA editing catalytic polypeptide. And this is important for defense against HIV-1, which we haven't talked about, but we will soon. So basically what this is, ApoBec3 is a cellular protein. It is it's a cytosine deaminase. It deaminates cytosines. So here it is, it will get packaged into the virus particle. So here's a HIV particle being made, and ApoBec3C is packaged into the particle. And then when this infects a new cell, so we have RNA, remember, is reverse transcribed to form a single strand of DNA, and eventually we have double-stranded DNA. But the enzyme ApoBec is going to do a cytidine deamination of C, and it's going to change it to a U. So now it will base pair with an A instead of a G. So you essentially introduce lots of mutations into the viral genome, and it's not infectious because you change a lot of C's. It's not infectious. So how does HIV exist, right? It exists because of a protein called VIF, VIF, right here, that little blue rectangle, which is one of the, so, the retroviruses mostly have GAG pol envelope, but HIV has GAG pol envelope and a bunch of little proteins. And one of them is VIF. They have accessory roles. What VIF does, it targets ApoBec to the proteasome for degradation. So the proteasome is the garbage disposal of the cell, chops up proteins. In order to do that, the protein has to be ubiquitinated. It has to be conjugated to a molecule called ubiquitin, shown as U here. And so basically, VIF forms part of this complex here that ends up ubiquitinating ApoBecs. So here's the three-dimensional structure of it. Here is the ApoBec. And VIF is part of this, linking all this together, and the ubiquitination enzyme is right there. So when ApoBec is degraded, obviously can't get into the virus particle, so we have normal reverse transcription. And so VIF is very important. If you take it out of the virus, it will not replicate. Now, VIF genes are shown on the right. These are genes encoding VIF. So humans and chimps have these, A, B, C, D, et cetera. So they've obviously been duplicated over evolutionary time. Some species have fewer. But look at the bats. This is one bat species. Has a lot of duplication of ApoBec. The implication is that bats are assaulted with viruses, so they need ApoBecs to protect them. It's quite interesting. All right, so that's an intrinsic defense. Another one is epigenetic silencing, which we've briefly mentioned here. Now remember, DNA in the nucleus is wrapped around chromatin, as you can see there. And unless the chromatin is modified, it's very tightly wrapped. It's compacted, and it cannot be transcribed effectively. And so chromatinizing a DNA is a good way to shut off its transcription. So when viral DNA gets in the nucleus, it's immediately chromatinized and it's silenced. Now, the way you get transcription is by modifying the histones. And here we're showing acetylation, which makes the DNA open and accessible to transcription. Now, viruses need to encode countermeasures to ubiquitination, right? Otherwise, they're not going to get going. So many viral genomes have them. Cytomegalovirus protein causes degradation of a cell protein called DAX, which is needed for histone deacetylation. So histone deacetylation would wrap the chromatin tightly and shut off transcription. So HCMV prevents that from happening. The Epstein-Barr virus, adenovirus proteins affect a protein called PML. So when viral DNAs go in the nucleus, these are nuclei of infected cells. They're staining blue with DAPI, and the green are centers of viral DNA synthesis. Those are, sorry, they're not centers of DNA synthesis. These are PML bodies, these green staining foci. When DNA comes in the nucleus, they're sent to PML bodies and chromatinized. So it's a way of inhibiting transcription. And these viruses prevent the formation of PML bodies. And the last one I want to mention again, retroviral DNA, when it comes in the nucleus, as soon as it integrates, remember, it does not get silenced when it's integrated. If it remains circular outside of the chromosome, it would be silenced. So integration is defense against epigenetic silencing. Apoptosis, programmed cell death. A virus infects a cell, the cell senses it, it undergoes death, programmed cell death. The idea being preventing virus reproduction so you spare the organism. Now, in the process of apoptosis, the cell releases what are called apoptotic bodies, and these are bits of the cell, and they may contain viral proteins, and so they are picked up by what we call sentinel cells. These include dendritic cells, macrophages, and NK cells, because they're going to figure out if there are foreign proteins in those apoptotic bodies. We'll come back to this later. Viruses can also regulate apoptosis, right? So that doesn't happen. And these are all the pathways leading to apoptosis. Here's apoptosis down at the bottom here, and all this stuff in the middle. And you don't need to know any of this, but the concept that receptors engaging ligands at the cell surface will trigger apoptosis. Okay, and there are three receptors: FAST, TRAIL, and TNFR1. So if you ever heard of tumor necrosis factor, tumor necrosis, right? It's killing tumors by inducing apoptosis. And viruses can antagonize that because TNF is produced in response to virus infection, and the viruses can counter. So it can counter the actual binding, it can counter the signaling that happens. It can also counter downstream events as well. And you can see all the other places where apoptosis is impaired or blocked. The mitochondria is also a point where apoptosis may begin. So from caspase 810, which is signaled from these three receptors, there's a signaling pathway through mitochondria that results in the release of cytochrome C, and that activates apoptosis here. But if the mitochondria is damaged in some way, it releases cytochrome C, and that will signal for apoptosis, because if you don't have mitochondria, you're really in big trouble. So some viruses damage mitochondria, and so the idea is that the release of cytochrome C is a evolutionarily conserved mechanism for inducing apoptosis. And then finally, the last intrinsic defense is CRISPR. This is ancient. It's in 90% of archaea and 50% of bacteria. It's not in eukaryotes, but now you know what it stands for. It is a way of remembering past viral infections or any invasion by DNA, by foreign DNA that comes in the cell. Here we have a phage injecting DNA. The DNA is chopped up by the CRISPR system, and little bits of the phage genome are integrated in the host DNA. Those are the palindromic repeats that were originally seen, and people wondered, what is this? And these are transcribed into CRISPR RNAs, short CRISPR RNAs. And then when a new phage infects that cell, the CRISPR RNA directs the degradation of the phage DNA by a Cas protein, a CRISPR-associated nuclease. So this is a defense system originally, and it was repurposed by scientists to do gene editing, right? But that's not its original purpose. In many clever ways, it was repurposed. But in bacteria and archaea, it is an intrinsic defense system. All right, let's see if we understand intrinsics. First question is, intrinsic defenses are always present. Which of the following are included? Antibodies, T cells, epigenetic silencing, skin, and mucus. All right, how did we do? Sorry, number 19. Yeah, it's epigenetic silencing. Intrinsic, not physical and chemical. Now, antibodies are adaptive later. T cells are later. Skin and mucus are physical and chemical barriers. Here we go. So anatomical and chemical barriers, skin and mucus. Then intrinsic would be silencing. Now let's talk about, for the rest of today, the innate immune system. That's our third barrier there, which can be activated within minutes to hours after infection. It's made up of cytokines, sentinel cells, which are dendritic cells, macrophages, and NK cells, and complement. Those are the components of the innate immune system. And the innate immune system collaborates with the adaptive system, and it will inform it if the infection is getting out of hand. So the innate immune system can, in fact, clear many infections. We're protected by it every day. We probably have viruses coming in, and the innate system protects us. But in cases where it doesn't work, then it informs the adaptive system. We'll talk about that next time. A big question in immunology was always, how does this innate system recognize other things and not itself, like viruses and not itself? So this was actually by work that wasn't even involving innate immunity. This was Nusslein, Volhard, and Wieschaus were working in 1980 in Germany to identify the genes of Drosophila that were involved in development. They were making mutants of flies and then letting them grow up. And they saw one fly that had a very unusual phenotype. And the legend is that Nusslein Volhard called over to Wieschaus. She was looking at it in the microscope and she said, "Das war ja toll." Wow, that's far, look at this, is far out, this phenotype. So they called them toll genes that encoded this for that reason only. It doesn't mean anything else. In 1996, they found that in flies, this plays some role in immunity also. So it's a developmental gene, but also immunity. And in 97, homologs or orthologs were found, homologs of the gene were found in mammals. And so that's how we discovered the sensors of the innate immune system through this fly gene. And today we know many, many sensors. So that was a toll-like receptor, but there are a whole bunch of others, and they're called pattern recognition receptors. And these are receptors, including the toll-like receptors, which you can see here. These are transmembrane proteins, but there are others as well. There are lectin receptors, there are nucleotide binding receptors, there are RNA binding receptors, even DNA binding receptors. And they recognize what we used to call PAMPs, pathogen-associated molecular pathogens. But in fact, it should be microbe-associated because not everything recognized is a pathogen. In fact, a lot of your gut microbiome, which is good, is recognized by these receptors as good and doesn't do anything. So it's not just pathogens. But the toll-like receptors can sense viral proteins on the cell surface, or from within endosomes, they can sense viral RNA. Let's take another look, a closer look at this pathway. So again, recognition of MAMPs. The cell surface TLRs can recognize viral proteins. And there are also a lot of TLRs in the endosome, so they can sense viral RNAs because the RNAs are passing through the endosome. And when they do sense these, when they sense the protein or nucleic acid, they initiate a signaling pathway, which is always a series of phosphorylation events. So for example, when TRR3 detects double-stranded RNA in the endosome, it activates a protein kinase called TRIF, which phosphorylates IRF3, which then dimerizes and goes in the nucleus, and it helps to activate the expression of downstream genes like interferons, interferon alpha and beta. So that's how all of these sensing pathways work, through a signal transduction pathway that ultimately turns on transcription of interferon genes in the nucleus. And on the right is the sensing pathway for cytosolic nucleic acids, including DNAs and RNAs of various sorts. They're sensed by two different proteins. The DNA sensing is on a separate slide. The two different proteins which activate a signaling pathway, and in the end, ends up putting transcription factors into the nucleus and turning on the transcription of interferon genes. So you can see this is the interferon beta promoter. It is responding to the presence of IRF3, 7, and F-kappa B, which have been allowed to enter the nucleus because they've been modified by phosphorylation in response to the sensing of nucleic acids in the cytosol. Now, these kinds of nucleic acids, 5' PPP, short double-stranded RNA, 5' PPP single-stranded, long double-stranded, these are not present in uninfected cells. So if they're present and sensed, that means there's a virus infection going on. Cells do not make those kinds of RNA, so that's how it's distinguished. DNA is also sensed. Now, I had some trouble with this for many years, but it's sensed in the nucleus. And most DNA viruses deliver their DNA into the, sorry, did I say nucleus? They're sensed in the cytoplasm. Most DNA viruses deliver their DNA to the nucleus. So I said, well, what's going on with sensing in the cytoplasm? Well, it turns out that everything in biology is leaky, right? So on the way in, some viruses break and they leave DNA in the cytoplasm. So that's when it's sensed. There are also sensors in the nucleus, which I'm not showing you. But this one is cool because if there's some DNA in the cytoplasm, double-stranded DNA, which is not supposed to be there in an uninfected cell, it is sensed by a protein called cGAS. cGAS is an enzyme, cyclic GAMP synthase. It takes a molecule of ATP and a molecule of GTP and makes a cyclic moiety of it, cGAMP it's called. And this is what it looks like. It's an AMP and a GMP with this unusual cyclic bridge. That molecule then is sensed by STING. STING is the police of DNA in the cytosol. Some of you got it. I wonder if they named it because of that. Stimulator of interferon. You see, they took two letters from stimulator. They did it on purpose. And then STING is normally in the ER, then moves with this cGAMP in it to the trans-Golgi, where it then initiates a signaling cascade. This TBK phosphorylates IRF3, goes in the nucleus, and turns on the transcription of cytokines like interferons. Okay, so that's how DNA is sensed in the nucleus. And viruses, of course, modulate sensing. They have to. This is a really efficient way of shutting down a virus infection. You'll see how the interferons work in a bit. I haven't told you that. But viruses make proteins that interfere at every sequence here. So here is a virus-infected cell with viral proteins in red, which are antagonizing the sensing. For example, let's see. Here is a herpes virus protein that is antagonizing the ubiquitination of RIG-I. RIG-I is one of the sensors of RNA. It needs to be ubiquitinated to work, and this viral protein is antagonizing that. These viral proteins are blocking the recognition of viral DNA by cGAS. And then hep C protein is blocking this STING complex, which results in phosphorylation of transcription factors and turning on type 1 interferon. So you get the idea that viral proteins have to interfere at multiple steps in sensing. Otherwise, interferon is going to be turned on and they're not going to do very well. And again, you don't have to memorize these. You just need to know this concept that nucleic acids can be sensed, in particular viral nucleic acids can be sensed, and the viruses produce proteins that block the sensing. This question, which of the following allowed the innate immune system to distinguish microbes from self? Cytoplasmic helicases and toll-like receptors. So cytoplasmic helicase is another word for RIG-I, one of the other sensors. Antibodies, apoptosis, ApoBec, all of the above. Hey, how do we do? Correct. A is the right answer. Cytoplasmic helicases and TLRs. So that allows the innate immune system to distinguish microbes from self. Antibodies do not. We haven't talked about them yet, but they attempt to eliminate the pathogen. ApoBec is an intrinsic, not innate, and it's not all of the above. So just these helicases. So let's take a look at those again. Again, these helicases, cytoplasmic helicases include RIG-I and MDA5, and then DNA sensors like CGAS. So the output of the innate immune system is cytokines, which includes interferons. And interferons are substances discovered in 1957. They found that if you took chicken cells in culture and exposed them to non-infectious influenza virus and waited a number of hours, the cell culture supernatant had something in it which, if you put on another set of cells, would block infection of influenza virus. So they say it interfered with infection of other cells, interferons. At the time, they didn't know what it was. And now we know that they're proteins. And they're produced by virus-infected cells, but also uninfected sentinel cells in response to products released from cells. So the virus-infected cells, we've just been talking about how when viral nucleic acid is present in a cell, it is sensed, and that leads to the production of interferon. So that's the virus-infected cell. The uninfected sentinel cell, how does that work? Well, here it is. Here is a mucosal epithelial layer with a basement membrane, right? Viruses are infecting them. And they're in the cell and they're being sensed. The cells are making cytokines in response to the innate system that we've been talking about. Those cytokines do a multiple things, including turning on other genes, but they also attract dendritic cells. And the dendritic cells will pick up these cytokines and go, oh, this is something going on here. And then these cells will apoptose, and the dendritic cell will pick up those apoptotic bodies and eventually bring them to the lymph node to see if there's something foreign in them, something we'll talk about next time. So both the infected cell and the uninfected sentinel cells make interferons. These sentinel cells will also not only respond to interferon, but they will make it as well. So interferons are of three types. There's type 1, type 2, and type 3. And there are receptors for all three types on cells. And they're shown here, the type 1, type 2, and type 3 interferon and their receptors. So here's type 1 interferon. That's one kind, interferon alpha. And that's the interferon type 1, type 2, type 1 receptor. And then we have type 2 interferon. There it is there, and its receptor, and then type lambda. And these receptors, as you may guess, are associated with kinases. These are all kinases, these proteins with weird names. So when the interferons bind, they turn on phosphorylation of these kinases, which initiate a signaling pathway. And that eventually turns on other genes. So here's the pathway of interferon transduction. So the production is rapid within hours of infection, and then goes back down again. Interferon, so here's a cell being infected by a virus. The RNA is sensed by the sensors. They initiate a signaling pathway that turns on interferon transcription. Interferon protein is made, gets out of the cell, and then goes and binds to interferon receptors on other cells. Okay, so it diffuses out of that first cell and goes and spreads to many other cells. It can go all throughout the body, in fact, because it can get into the blood. This interaction of interferon with its receptor also initiates a signaling pathway, which turns on transcription of what are called ISGs, interferon-stimulated genes. So there are like thousands of them, and they do different things in different tissues. So most of them we don't understand how they're working, in fact. So those are the interferon-stimulated genes, over 1,000 cell proteins, and those have antiviral activity. You see this ISG is breaking up a viral capsid. Interferon itself has no direct antiviral activity. It's the interferon-stimulated genes and their encoded proteins that have the antiviral activity. So interferon is used as a treatment for some virus infections. It's not the interferon that's treating the infection, it's the genes induced by it. The promoters that run these ISGs, some of them come from retroviruses. They're parts of endogenous retrovirus LTRs. There are a bunch of LTRs that contain motifs that respond to interferons, and they occupy about 6 to 14% of the mammalian genome. And so what has happened is, remember, hosts have been infected years ago, many, many years ago, by retroviruses. The DNA integrates into the host germ cell, so it's passed on. And then you have this sort of arrangement here where you have the endogenous retrovirus. And then the LTRs are repurposed. They're moved around elsewhere in the genome to serve as promoters for interferon-stimulated genes. So another example of how we are using these. And these are, this has happened in multiple lineages, not just us, but many other animals as well. So the boxes indicate when this usage of the LTR occurred. Another interferon, I'm going to go through a few interferon-stimulated genes just to show you how they work. One of them is called tetherin, which works exactly as it might sound. It tethers the HIV particle to the cell surface. So tetherin is this molecule that's shown here, which bridges the plasma membrane and the viral membrane. And so it actually binds to the virus in the endosome. And then when the virus particle tries to bud, it cannot, because they're all linked together with these tetherins. And this is an electron micrograph of what happens in cells. You can see the virus particles. This is a cell at the bottom here. And the virus particles are all stuck because of tetherin, and they make chains and never get away from the cell, so they're prevented from spreading. Now, you may say, how does HIV exist with tetherin? Just like ApoBec, right? VPU is another one of these small viral proteins. And in this case, it's a tetherin antagonist. So VPU prevents tetherin from working. Otherwise, the virus wouldn't exist. Some other interferon-induced proteins that I like, this one's called IFIT1. This protein binds RNAs lacking 2'O methylation. What is 2'O methylation? So here is the cap on an mRNA. So there's the G, the cap G, and then 3 phosphates, and then the first base of the mRNA. Along with the cap, remember, these bases are methylated. That's part of the cap function. If they're not present, IFIT binds to it and prevents interaction of the cap binding protein and with the 5' end with the cap, and prevents translation. The idea being that if there are no methyls, then it must be foreign, because our transcriptional machinery puts methyls there. And on the right are 2 structures that illustrate that are very nice. Here is the structure of, here's the structure of IFIT1, and this is the cap fitting inside of it, that red blob. And on the right is a cross-section of the solid structure, and you can see it's binding the cap with the little red dot there, and then a few other bases of the RNA. So basically, it blocks the RNA from being translated. But many viruses escape from IFIT in many ways. Like flu does, it steals the caps for its mRNAs during transcription, during RNA synthesis. You may not remember that, but the very 5' end of all influenza mRNAs come from the cell, in which case they have those methylations present. So IFIT doesn't bind it. Coronaviruses don't have a cap. They have a protein stuck on the 5' end. IFIT doesn't bind it. Some viruses encode a methylase. So they actually put a methyl group on those bases of the cap. So they're not affected by IFIT. And some viruses steal host enzymes to do that. And finally, in some viruses, there's an RNA structure right near the cap, and that blocks IFIT from binding. So lots of ways to get around this. Back and forth, as Alice might say. This is another cool one, IFIT M3. It inhibits fusion. So IFIT M3 is this protein shown on the right here. It is so strange. So it has a transmembrane domain. That's the phospholipid bilayer of the cell. It's a transmembrane domain. And then it has some lipids attached to it, which stick in the membrane. And there's another part of the protein here, this orange part, which disrupts the membrane and prevents fusion. And so, remember, viruses can fuse at the cell surface or from within endosomes, and IFIT M3 will block it. And here's how it works. Here's a diagram of it. So we have the virus and the host membrane. And remember, the receptor-virus interaction is going to bring these together, and you're going to get fusion. But these IFITs disrupt the membrane topology and block fusion. So the second fusion cannot occur, and so the viral nucleic acid never exits. So I think that's a pretty cool strategy for doing that. I don't know how antagonism works. Nobody's figured that out yet, but there must be one. Now, the interferon system is dangerous. Even though it has these good effects, it's dangerous. When you get a virus infection, we say you have flu-like symptoms, even though you may not have flu, because of the interferon inducing all these gene products, because most of our cells have interferon receptors. So if you have a local infection and interferon is made, it's going to diffuse throughout your body and give you fever, chills, nausea, malaise, muscle aches, etc. We call that flu-like symptoms because they're actually interferon-induced symptoms, but that's what we call them. And almost every viral infection results in interferon production. So we say you have flu-like symptoms, but you might not have flu. You could have any other virus infection. And when you treat people with interferon for a virus infection, for years, hepatitis C virus infection was treated with interferon. The patients feel horrible because you're inducing all these interferon-stimulated genes. They get fever, chills, nausea, and malaise all the time. So they stop taking the interferon because they feel so badly. We don't do that anymore. We have great antivirals that are much, much better. Now, early in COVID, we tried to do it, but didn't work very well. Last question. How do interferons limit virus replication? Interferons directly inhibit viral translation. Interferons lyse viral particles. Interferons induce ISGs. Interferons damage cells. None of the above. So the next time you have flu-like symptoms, you will realize it's caused by interferon. Even getting a vaccine can give you some flu-like symptoms, right? Because the vaccine antigens are sensed by the innate immune system. So you get pain at the injection site, you may have some fever. That's good. It means that the vaccine is being sensed. And exactly why that is, you'll see in a bit. Okay, how did we do? Interferons induce ISGs, that's right. They do not directly inhibit anything. Right? They always induce ISGs, which then inhibit something. How about these sentinels? What are they doing? Remember what they are, dendritic cells, macrophages, natural killer cells. So right now, as you are sitting here, these cells are flowing all through you, all through your fluids and your tissues, and looking for signs of change to see if you're infected. So that's why we call them sentinels. So here are some dendritic cells cozying up to other cells. They get right up to cells. And here are some NK cells also. The NK cell is orange, and getting up to another cell. How they work, you'll see in a minute. Now, dendritic cells are very cool. There are all kinds of dendritic cells. There are blood dendritic cells, there are tissue with different markers. You don't need to know any of that. All you need to know is that they patrol throughout your body, and there are specific ones in your skin, in your intestine, and so forth, in your other tissues. So here would be epithelial cells, skin, eye, vagina, for example, and the green are the dendritic cells. Here is a virus infection, so the dendritic cells are grabbing virus particles or virus proteins, and they're going to bring them to the local lymph node. So if you have an upper tract infection, which would be nasopharynx, your lymph nodes in your head are going to be sampling them, not in your feet. Doesn't make any sense, right? The local ones, and that's why there's two lymph nodes right under your throat here. You can feel them, and when you get a cold, they swell. It's a great sign of having a cold. They're swelling because of things we're going to talk about, initiated by dendritic cells going in to the lymph node, which contains lots of T cells, and they will present what they have found and say, is this foreign? So here is actually a beautiful fluorescence photograph of a lymph node. There are some blood vessels in red. The T cells are in blue, and you can see this dendritic cell with all its processes snuggling up to a T cell. It's presenting an antigen that it's picked up in its journeys. It does this all the time, even if you don't have an infection. Picks up proteins and brings them in and says, is this a problem? That's how it works. Same thing happens in the gut. There are different kinds of dendritic cells, but they all end up in the lymph node as well. This one you can see in purple is sticking one process through the intestinal epithelium into the lumen of your gut, where lots of food is, lots of antigen. They're just sampling it to see if there's anything bad in there. Okay, so that's what dendritic cells do. And the series of events are summarized here. We have an infection of an epithelial cell. The cells sense the virus. They make cytokines. The cytokines attract sentinels. Here we're showing a dendritic cell. And these dendritic cells can also be activated by proteins or nucleic acid released from infected cells or apoptotic bodies that are taken up by the dendritic cells. When they are given these gifts, they get activated. They become a mature dendritic cell. Then they go into the lymph node where they interact with T cells. So here's what happens to these dendritic cells. They're present initially in an immature form. They have receptors for everything. They have toll-like receptors. They have cytosolic RNA receptors. They can take up apoptotic cell bodies. They have receptors for cytokines. They have MHC class II in the endosome, which is going to display the antigen eventually. They can make interferon themselves. So when they're up against that epithelial barrier and they're sensing interferon produced, they can make more to try and help clear the infection. Now, when they get matured and activated, because they're given an antigen, they look like this. They have these typical processes, which is why they're originally called dendritic cells. The MHC is now put on the plasma membrane, carrying, which is shown in orange, whatever antigen it has found. And we'll talk about the antigen processing pathway next time. And what it's doing here, it's now in the lymph node, and it's presenting this antigen to a naive T cell which has never seen antigen before. And this peptide is presented to the T cell receptor from the MHC2 molecule. MHC2 is unique to these sentinel cells. And then the dendritic cells is also giving cytokines of various sort to the T cell. And then if this is foreign, the T cell becomes activated. Foreign means it's recognized by a T cell because during development, all the T cells that recognize self are supposed to be eliminated in the thymus. So if you have a T cell that reacts with an MHC-presented peptide, that means it's foreign. In principle, it doesn't always work right. You get autoimmunity and so forth, but that's the idea. Then that activated T cell will go on and get involved in making antibodies and T cell immunity. And then finally, NK cells, well, not finally, but second is NK cells. These recognize cells that are lacking MHC. And if there's no MHC, because many viruses downregulate MHC from the cell surface. So the NK cell said, aha, this cell doesn't have MHC, I'm going to kill it. So on the top, we see an NK cell and a target cell, which is infected. The virus has downregulated the MHC, 2, or 1 in this case, because this is just some epithelial cell. And it's going to kill it. So it has this activating receptor, which binds to another protein on the target cell, and it will kill the cell. But if there's an MHC molecule present, the NK cell assumes that there's no virus infection and it leaves it alone. But you know what? Viruses can make their own MHC. Sneaky, right? And the NK cell thinks it's normal and so it doesn't kill the cell. So those are modulators of this NK killing. All right, now finally, complement, which is the other component of the innate system, which is this horribly complex set of proteins that sense different things and end up killing cells, essentially. So but here you have these proteins circulating in your blood all the time. And for example, if you have antibodies binding a microbe or a virus or a bacterium, a part of the complement system, this C1, will bind those antibodies and stimulate the production of all these proteins that lead to inflammation, right? That's the production of cytokines. Then we have other things that are sensed, like carbohydrates, and these can lead to production of pores in membranes that will lyse the membrane of whether it's a virus or a bacterium. So a very effective system that can act alongside the innate immune system. Well, it's part of it. And yes, there are viral modulators of complement as well. So basically what we've just talked about is that when you get infected with a virus, you get inflammation. So what is inflammation? It is the production of cytokines and chemokines that result in redness, pain, heat, and swelling. These are the 4 classic signs of inflammation. In Latin, they are rubor, dolor, calor, and tumor, originally described by this Roman medical person, Celsus, in the 1st century. He noticed patients having these symptoms. He didn't know why, but he gave them names. And inflammation is not just caused by viruses. This is a picture I pulled off the internet. Cancer, cardiovascular diseases, Alzheimer's, pulmonary, arthritis, autoimmune, neurological diseases, diabetes, they can all cause inflammation. When you go running and you twist a muscle or a tendon, the healing process has inflammation associated with it. By the way, what's missing in that picture? Viruses. Viruses and bacteria cause inflammation too. So what is this redness, pain, heat, and swelling? So again, these are caused by the cytokines and chemokines induced by the innate immune system. That they represent increased blood flow, increased capillary permeability. So the capillaries get permeable so that the immune cells can get out of them into surrounding tissues and work on the infected area. And influx of phagocytic cells, so lots of macrophages go into the area to try and clear up. So that gives you swelling and tissue damage caused by all of this activity. So that is what we mean by inflammation. And infection causes that. And these are some of the players. We have cytokines that are involved in inflammation. There are 3 groups. There are some that we call pro-inflammatory, like famously TNF, IL-6, IL-12. So the late phase of COVID involves an inflammatory phase. And one of the therapies was giving patients an antibody to IL-6, because this is driving inflammation. When I mean it's driving inflammation, it's recruiting it's causing all these activities that we listed in the previous slide. Then any pro-inflammatory cytokines have to be reversed, so we make anti-inflammatory cytokines to turn it off. So they suppress the pro-inflammatory cytokines. And then the chemokines recruit immune cells to the infected area. All right, so here's a diagram of how this is happening. We have some tissues that are infected. They're making chemokines. They're attracting phagocytic cells. Here are some neutrophils coming in. And they're coming in from the blood. But all of this is undergoing inflammation. It's swelling due to the presence of increased fluids and phagocytic cells and the permeability of the capillaries. So these cytokines that are made function locally. So here we have a local infection. They function locally, but they can get into the circulation. You can see they can easily diffuse into the blood. So they get everywhere and have global effects. That's what I've been telling you so far, like sleepiness, lethargy, muscle pain, no appetite, nausea, many others. Flu-like symptoms. So even if you have a localized virus infection to your upper tract, to your nasopharynx, you may feel nausea, which there's no virus replicating in your gut. It's the cytokines and chemokines that are giving that feeling. Here's an example of how cytokines work. On the left, we have a cytokine binding its receptor. It initiates a signaling pathway. And here you can see there's some proteins going in the nucleus and turning on the transcription of genes that eventually give a response, whatever the cytokine or chemokine is doing. And on the right is an example of that. Here are lymphocytes that are going through the blood cell wall, the endothelium. So if there's a localized infection, for nasopharynx, for example, there are capillaries there that will bring phagocytic cells to the site, and then they will squeeze through the gaps between the endothelial cells. This process is mediated by chemokines and their receptors. So here it's shown at the bottom here. You see when the lymphocyte first hits the wall, it's actually binding to receptors. And then it starts to roll and it activates other receptors and then it goes through. And so here's what happens here. These are all the interactions that occur. A chemokine produced by the endothelial cells is inducing receptors on the surface that are involved in attachment, as you can see here, as are other receptors as well. So this is a complicated series of events induced by chemokines and cytokines. That's what they're doing. And of course, viruses counter these. They have to because they're all very effective. We have countermeasures where the production of cytokines are interfered with, the synthesis. And these are cases where the viral protein interferes with cytokine action. The virus encodes homologs to block receptors. So the virus will encode what looks like a cytokine. It will bind to a receptor on the cell. It doesn't work the same way, so you're not going to get any signaling. It's going to interfere with its function. And there are also antagonists that alter the signaling pathway that I just showed you. And then there are some countermeasures that inhibit the production of functional cytokines. And there are some that encode, some viral genes that encode soluble cytokine receptors. They bind the cytokines and then nothing happens because these are soluble cytokine receptors. They're not on a plasma membrane, which is what you need in order for signaling to occur. So every step of infection involves some attack and counterattack. Inflammation is actually good for you. It stimulates a really good adaptive response. And one of the reasons cytopathic viruses induce good immune responses is that they cause inflammation. They kill cells. You have pieces of cells floating around that can be picked up by sentinel cells, the innate response, and collaborate with the adaptive response. So cytopathic viruses are very good at stimulating immune responses for that reason. They cause inflammation. Now, to counter this, the genomes of cytopathic viruses often encode proteins to counter inflammation. I've told you a lot about this already. Right? They have to counter the induction of inflammation. Otherwise, you're going to get a great immune response and the virus is going to be cleared. So herpes viruses, adenoviruses, pox viruses, all are cytopathic viruses that encode inhibitors of inflammation. Right, that's cytopathic viruses. But many viruses do not stimulate inflammation. And these are what we call non-cytopathic viruses. Some viruses do not kill cells. There's no apoptosis, there is no necrosis. So there are no bits of cells coming off that the sentinels can pick up. And consequently, they have nothing to say to the adaptive response. So there's very little collaboration between the innate and adaptive. So you get a poor immune response to viruses that do not kill cells. They cause often persistent infections because they can't be cleared. There's a lousy adaptive response, all because they don't kill cells and they don't induce a good inflammatory response. All right, so we're going to talk about acute and persistent infections later. The acute infections are done by viruses that kill cells and initiate a great adaptive response, and the persistent infections are ones that do not. So here's the lesson from this. The classic inflammatory response, this thing, heat, redness, swelling, pain, is telling you that the innate and the adaptive responses are communicating. If there's no inflammation, they're not communicating. No inflammation would be in a virus that does not kill cells. So you're going to get a bad adaptive response, which means not good antibodies and T cells, and you're likely to have a persistent infection. Now, many vaccines do not replicate, right? So they can't kill cells. They can't make a great adaptive response because they don't cause inflammation. So we try to get around that by putting adjuvants in many vaccines. Adjuvants stimulate an innate immune response. They work in other ways too, but one of the ways they work, for example, they can bind to toll-like receptors and turn on the synthesis of cytokines and chemokines, cause inflammation. And again, that's why you get inflammation at the injection site, because you have some kind of adjuvant in the vaccine. The mRNA vaccines for COVID, the lipids in those vaccines, remember there are mRNAs in lipid nanoparticles. Those were adjuvants that caused good inflammation. Now, I told you already, many things can cause inflammation, not just infection. And if you scratch your skin, right, you get inflammation. You've noticed it. You have your cut, and then it starts to heal. It gets all red around it. It gets swollen. That's inflammation. And here's an example of an injury that benefits vaccine efficacy. So the smallpox vaccine was delivered. We still use it in certain populations. Now that the virus is eradicated, not as much. It used to be delivered in this bifurcated needle. There was a drop of vaccine in it and very sharp tips, and they would take the vaccine-containing needle and scrape your deltoid here and basically make cuts in your skin. The virus would then replicate there and make a good antibody and immune response. But it turns out the scraping induces inflammation. And that's a big part of the efficacy of the smallpox vaccine, because if you inject the smallpox vaccine, it doesn't work as well. You need this damage, which causes cells that are damaged to release chemokines and cytokines, which recruit immune cells to the area, and that boosts the immune response. So it's inflammation caused by the vaccine delivery method, and it benefits vaccination. We didn't actually know this. We just found that this method is called scarification, is better than injecting. So we stuck with it. And then a few years ago, a study was done showing that it's the injury caused by scarification that gives you a great immune response because of inflammation. It was great. I love it. Okay, remember one thing here today. All viruses need to encode at least one regulator of intrinsic and innate defenses. And this includes sensing, interferon production, signal transduction, cytokines, chemokines, NK cells, DCs, dendritic cells, and complement. Every virus has to have multiple antagonists, otherwise they get eliminated. These are such effective systems. Today, we finished with the innate system today. So we've made cytokines, we've got inflammation happening. Next time, we're going to talk about how that turns on the adaptive system, and we make antibodies and T cells.